Welcome to edupediaworld.com. This is Somjaja Nayak, your online biology tutor. In this video, we are going to discuss about regulation of gene expression. Regulation of gene expression. Regulation of gene expression refers to a very broad term that may occur at various levels. Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be exerted at transcription level, that is the formation of primary transcript, the processing level or regulation of splicing, transport of mRNA from nucleus to the cytoplasm, and the translational level. Regulation in beta-galactosidase gene in E.Coli. The gene in a cell are expressed to perform a particular function or a set of functions. For example, if an enzyme called beta-galactosidase is synthesized by E.Coli, it is used to catalyze the hydrolysis of a disaccharide lactose into galactose and glucose. The bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of the enzyme beta-galactosidase. Therefore, in simple terms, it is the metabolic, physiological or environmental conditions that regulate the expression of genes. The development and differentiation of embryo into adult organisms are also a result of the coordinated regulation of expression of several sets of genes. Regulation in prokaryotes. In prokaryotes, control of the rate of transcriptional initiation is the predominant site for control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affect its ability to recognize the start sites. These regulatory proteins can act both positively, that is activators, and negatively, that is like repressors. The accessibility of the promoter regions of the prokaryotic DNA is in many cases regulated by the interaction of proteins with sequences termed operators. The operator region is adjacent to the promoter elements in most of the operons, and in most cases the sequences of the operator binds a repressor protein. Each operon has its specific operator and specific repressor. For example, Lac operator is present only in the Lac Operon and it interacts specifically with the Lac repressor only. Now you will have a small doubt. What is an operon? An operon is a functioning unit of genomic DNA containing a cluster of genes under the control of a single promoter. And the Lac Operon is an example for that. Now we will discuss about the Lac Operon. The elucidation of the Lac Operon was also a result of a close association between a genetist Francois Jacob and biochemist Jacob Monod. They are the first to elucidate a transcriptionally regulated system. Jacob and Monod are awarded the Nobel Prize for their work. In Lac Operon, here the Lac refers to lactose. A polycistronic structural gene is regulated by a common promoter and regulator genes. Such arrangement is very common in bacteria and is referred to as operons. The Lac Operon consists of one regulatory gene, the I gene. Here the term I does not refer to inducer, rather it is derived from the word inhibitor, and three structural genes Z, Y and A. The I gene codes for the repressor of the Lac Operon. The Z gene codes for beta-galactosidase, which is primarily responsible for the hydrolysis of disaccharide lactose into its monomeric units like galactose and glucose. The Y gene codes for permease, which increase the permeability of the cell to beta-galactosides. The A gene codes a trans-acetylase. Hence, all the three genes product in Lac Operon are required for the metabolism of lactose. In most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. Now you will have a doubt how the Lac Operon is working. Lactose is the substrate for the enzyme beta-galactosidase and it regulates switching on and off of the operon. Hence, it is termed as inducer. In the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cells through the action of permease. Remember that a very low level of expression of Lac Operon has to be present in the cell all the time. Otherwise, lactose cannot enter the cell. The lactose then induces the operon in a following manner. The repressor of the operon is synthesized, which is all time constantly, from the I gene. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. In the presence of an inducer such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. Essentially, regulation of Lac Operon can also be visualized as regulation of enzyme synthesis by its substrate. Regulation of Lac Operon by repressor is referred to as a negative regulation. Lac Operon is under the control of positive regulation as well, but it is beyond the scope of discussion at this level. This is a diagrammatic representation of how the Lac Operon is working. In the first part, you can see that in the absence of an inducer, the repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. Then, in the presence of an inducer, such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. This allows the RNA polymerase access to the promoter and the transcription is proceeding. The Human Genome Project. In the preceding sections, you have learned that it is the sequence of bases in DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequences. If two individuals differ, then their DNA sequences should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques where it was possible to isolate and clone any piece of DNA and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. The Human Genome Project was the international collaborative research program whose goal was the complete mapping and understanding of all genes of the human beings. All our genes together are known as our genome. Human Genome Project or HGP was called a mega project. Human genome is said to have approximately 3 into 10 raised to 9 base pair. And if the cost of sequencing required is $3 per base pair, then the estimated cost in the beginning or the total estimated cost of the project would be approximately $9 billion. The goals of the Human Genome Project. The first one is to identify all the approximately 20,000 to 25,000 genes in human DNA and to determine the sequences of 3 billion chemical base pairs that make up the human DNA. The third one is to store this information in some databases. Then to improve the tools for the data analysis. Then to transfer the related technologies to other sectors such as industries. And to address the ethical, legal and social issues ELSI that may arise from the project. The Human Genome Project continuation. The Human Genome Project was a 13-year project coordinated by the US Department of Energy and the National Institute of Health. During the early years of the Human Genome Project, the Wellcome Trust or the UK became a major partner. Additional contributions came from Japan, France, Germany, China and others. The project was completed in 2003. Knowledge about the effect of DNA variations among individuals can lead to the revolutionary new way to diagnose, treat and someday prevent the thousands of disorders that affect human beings. Methodologies. The methods involved two major approaches. One approach focused on identifying all genes that expressed as RNA, that is referred to as expressed sequence tags. The other took the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequences and later assigning the different regions in the sequence with functions, a term referred to as sequence annotation. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes. That we call DNA is a very long polymer and there are technical limitations in sequencing very long pieces of DNA and cloned in suitable host during specialized vectors. The cloning resulted into amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used hosts were bacteria and yeast, and the vectors are called as BAC or bacterial artificial chromosomes and yeast artificial chromosomes. The fragments were then sequenced using automated DNA sequencers that worked on the principle of a method developed by Frederick Sanger. Salient features of human genome. Some of the salient observations drawn from Human Genome Project are as follows. The human genome contains 3,164.7 million nucleotide bases. The average gene consists of 3,000 bases, but sizes vary greatly, with largest non-human gene being dystrophin at 2.4 million bases. The total number of genes is estimated at 30,000, much lower than the previous estimates of 80,000 to 1,40,000 genes. Almost all, that is the 99.9% nucleotide bases are exactly the same in all people. The functions are unknown for over 50% of discovered genes. Less than 2% of genome codes for protein. Repeated sequences make up very large portion of the human genome. Then repetitive sequences and structures of DNA sequences that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics and evolution. Then chromosome 1 has the most genes, 2,968, and the Y has the fewest, 231. And the last one, scientists have identified about 1.4 million locations where single base DNA differences or SNPs or single nucleotide polymorphisms, that is produce SNPs, occurs in human. The information promises to revolutionize the process of finding chromosomal locations for disease associated sequences and tracing the human history. Applications and future challenges. Driving the meaningful knowledge from DNA sequences will define research through the coming decades, leading to our understanding of the biological systems. This enormous task will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both the public and private sectors worldwide. One of the greatest impacts of having human genome sequence may well be enabling a radically new approach to biological research. In the past, researchers studied one or a few genes at a time. With the whole genome sequences and a new high-throughput technologies, we can approach questions systematically and on a much broader scale. They can study all the genes in a genome, for example, all the transcripts in a particular tissue or organ or tumor, or how tens of thousands of genes and proteins work together in interconnected network to orchestrate the chemistry of life. The DNA fingerprinting. DNA fingerprinting, also called DNA typing, DNA profiling, genetic fingerprinting, genotyping, or identity testing in genetics. That is a method of isolating and identifying variable elements within the base pair sequences of the DNA. The technique was developed in 1984 by British genetist Alex Jeffreys, and after he noticed that certain sequences of highly variable DNA, known as mini satellites, which do not contribute to the function of genes, are repeated within the genes. Jeffreys recognized that each individual has a unique pattern of mini satellites, the only exceptions being multiple individuals from a single zygote such as identical twins. The procedure for DNA fingerprinting. First, obtaining a sample of cells such as skin, hair or blood cells which contain DNA. Then the isolation of DNA, digestion of DNA by restriction endonucleases, then separation of DNA fragments by electrophoresis, transferring or blotting of separated DNA fragments into synthetic membranes such as nitrocellulose or nylon, then hybridization using labeled VNTR probe, and the detection of hybridized DNA fragments by autoradiography. This is a schematic representation of DNA fingerprinting. Here I am giving one example also. From a crime scene, investigators got some samples. They have two suspects. So they did a DNA fingerprinting to identify who is the actual criminal. Few representative chromosomes have been shown to contain different copy number of VNTR. For the sake of understanding, different color schemes have been used to trace the origin of each band in the gene. The two alleles, the paternal and maternal of chromosome, also contain different copy numbers of VNTR. It is clear that the banding pattern of DNA from the crime scene matches with the individual B and not with the individual A. And in addition to the application in forensic science, it has much wider applications such as in determining population and genetic diversities. Currently, many different probes are used to generate DNA fingerprints. Thank you. In the next video, we will be moving to the next chapter, the evolution.